He's a neuroscientist and an electrophysiologist working on the development of engineered hybrid biological systems for functional drug screening, toxin detection, lab on a chip, and robotics applications. Dr. Molnar has more than 30 publications, and he's now an assistant professor at the University of Central Florida. Dr. Molnar, please.
Thank you very much for the invitation and the opportunity to present my work here. And it is a real honor for me to be in here and talk to you. I would like to talk about in vitro systems. And I don't know how my topics fits in the topics of the conference, which is nanomedicine and telemedicine. But as I was thinking about it, I do believe that there are many connections to what I am doing and what other people is doing in here. So let's step over and see what I will be talking about. So first I would like to sketch to you the future or my dreams, so long-term plans, what I would like to do. What I would like to do is treat biology as components and develop tools to manipulate cells. Yesterday we heard a presentation who was talking about creating tools to manipulate atoms and build robots. I would like to create tools to manipulate cells, biological components, and build systems out of them. Maybe not robots, but at least functional systems which can be used for something. So I would like to discover those possible interaction with biological systems. For me, these are cells which can be used to manipulate the cells and let them do what I would like them to do. Of course, these tools are based on biology and cannot devote for the built-in capacity of the biological component. So my task is to learn from biology and apply this knowledge to manipulate it. It's not new, but I'm trying to put it in a new view. And so what I can use this system for? One of the ideas which is connected to the topics of this conference is individualized medical care. The idea for telemetry is to take some samples from a patient, maybe do a genetic analysis. If it's not too conclusive, then set up the patient with telemetric sensors and observe the reaction of the patients to different drugs. That is what I would like to do in vitro. So take a cell from the patient, create a functional system with that cell. This cell could be a stem cell, hopefully can be differentiated to different type of cells of the body. So I have a hope to take a cell from a patient, put it in a system, create a functional test system, and screen drugs on it, and select that drug which is the most appropriate for that patient. So it's a different approach for the same question. The other possibility is for complement traditional pharmaceutical screening. At this moment, there are serious problems with screening drugs in pharmacology. One of the problems is that most of the models are animal models, and animal results not always translate to human results. So it would be much more beneficial if we could screen drugs on humans. Of course, it's not possible, so let's test drugs on human cells in a functional system. Functionality is important for me. Traditional pharmaceutical screening is done on single receptors, on single targets. I do see a significance to do screening on complex system, not in vivo, but built, engineered complex functional biological system built from human cells and run screening in a high throughput way. And also, I see a connection to prosthetics. If we learn how to, I'm working with the nervous system, so I'm thinking about connecting, for example, robotic arms to the nervous system. So if we learn how to persuade the biology to connect to our electronic device, instead of sticking electrodes, simple electrode in it, and evoke unwanted biological reactions, then we may be able to integrate seamlessly our electronic device with the biological system. So also I see connections to robotics. Neurocomputing, it's an interesting field, how the nervous system is processing information, how can we create an interface to the nervous system. So this is also my topics. So these were the dreams. These were the dreams. What are reality? Let's talk about it a bit. And what are the problems with this view? What I would like to do is to build biological systems. In engineering, it's easy, more or less. So if we have a clock, we are taking apart, we are studying parts, and after it, we are putting together, and it works. In biology, we have a chicken, we are dissociated to cells, we will not be able to put together the chicken again. So there is a big difference. How can we approach biology? And basically, the classic engineering principles are not applied. What tools we have to recreate complex systems from biological components? The trivial, commonly used method is modeling. We can study the components, for example, ion channels, receptors, create a mathematical simulation, and we can simulate how the whole system is working. What I would like to do is trying to reorganize the components, the cells again, and create a functional working biological system. So I see these two tools. We noted that novel engineering principles and tools needed to manipulate biology. For me, self-assembly is this tool. So instead of the classic method to take single cells and put them together, or take single atoms and put them together, I trust in self-assembly. And I do believe that biology has the capacity, which is obvious for self-assembly, if we are able to find the appropriate clues and appropriate conditions to determine where the self-assembly will lead, or what is the outcome of the self-assembly. So every cell has an internal program. It's interacting with the environment, and every cell is doing something. Question is that basically the extracellular clues are activating the cell internal program. Our problem is at this moment we have no idea what is this internal program, or we have some ideas, but usually they are false. So we have a problem, what is the internal program, so what the cells can do. We have a problem knowing what the status of the cell, because this program has a very nice memory. It depends what we have done with the cells, and we don't know that. And also we don't know what are the extracellular clues to activate this program. So we don't know a lot of things, and we have a very limited knowledge and very limited tools at this moment to manipulate the cells. So the other problem is technical. I would like to say that in this project, technical problems are less than our limited knowledge about biology, but there are very serious technical problems. The technical problem is that we have to present to the cells in a extracellular clues in a time and spatial dependent manner. And this question is very similar to nanomedicine. We are talking about chemical programming that sensing the state of the cell and release a different drug, and depending what is happening, release a same problem. We have a cell which is doing something, and we have to give the cell the appropriate signal to the appropriate time, which is not really solved, and I do believe that even in stem cell research, it's a very serious problem. So this is an unsolved technical problem. So just a sketch, what we would like to do. We have a cell, it has an integral program, it's communicating with the environment, and it is doing their internal program according to the signals, what it is receiving, what are the signals we can apply to a cell. We can modify surface contact interaction signals, and we can modify the extracellular solution and give signals through the solution. Manipulating the solution where the cells are in, it's an easy thing. Changing the medium, putting in growth factors, it is done for a long, long time. What is the problem is, which is surprising, that we're still doing most of our cell culture work in serum containing medium with 20,000 unknown protein in it. So this statement that we know what the cells need for growing and for doing their job, it's sometimes false. So problem is that we have to formulate the environment of the cells very carefully, and we just don't have the appropriate knowledge for that. So I put in one more slide about neuronal engineering. So what about, this is connecting external devices to the nervous system. What are the problems there? And I would say biology is just incompatible with silicon-based electronics in any aspects. This incompatibility coming from the materials we are using. We are using solid materials like silicon, metal. The biology is using soft materials like hydrogels. And the Young modulus and the mechanical properties are different. Maybe this problem will be solved by the nanomedicine. This problem is not coming up if you are using 100 nanometer sized robots. But in prosthetics, anything what we would like to do now with biology, it's a problem. Also, biological reactions is a serious problem. Anything we are putting in the body, the biology is reacting to it and do something what we don't expect or we are not able to handle in many cases. If you are implanting electrodes, the electrodes are encapsulating, so biocompatibility or changing the biological reaction is important. That is also can be the outcome of my study, in vitro studies, to come up with ways to change the reaction of the biological system to the man-made materials. And also, I'm not talking about data interpretation and computing. The nervous system is just different. The first topics I would like to talk is to create engineered neuronal systems. So I would like to have neurons. I would like to determine how those neurons are interconnected. I would like to put them in a test system, and I would like to screen drugs which are acting on the nervous system and acting not on single cells, but on a complex phenomena like synaptic transmission or long-term potentiation, which is a memory model. I would like to have this test system relatively high throughput, so I don't want to stick electrodes in cells in the classical electrophysiological way. I would like to do extracellular recording, which is non-invasive and relatively high throughput. What can do at this moment? We are able to determine attachment of cells, and we are able to determine axonal growth or process growth of the cells. How can we do it? We can do it by manipulating the surface properties of the culture surface. In this example, we are using two chemicals. We are using self-assembled monolayers. We are using two chemicals, one of them hydrophile, the other one hydrophobe. With photolithography, we are able to create a two-color surface, which means some parts of them is hydrophile, some parts of them hydrophobe. If we are plating cells on them, the cells will sit on the hydrophile surface and send processes on the hydrophile surface, and they just don't grow on the hydrophobe part. So this example, this is the photomask we are using. We have sites for cell attachment. We have interconnectivity pathways for the cell. We have some speed bumps, which is preventing the process of this cell grow in this direction. And if we are plating cells on them, in high probability, they will be finding the cell attachment site, and they are sending processes on the predetermined pathways. In this example, I just filled up the cells with fluorescent dyes with patch pipettes. I did 2 cell patch clamp recordings on them. What can we see? We can see that these cells are physiological. That means they are neurons. They have voltage-dependent ion channels. They are generating action potentials. And also, we can see that if I am evoking an action potential in one cell, I get inward current in the other cell, which means they are interconnected. So they are behaving normally like they would behave in a cell culture. I wouldn't say completely that they would behave like in vivo, because it's always a debate how good an in vitro system is modeling an in vivo system. But these are hippocampal cells from rat. These cells behaving pretty much the same way as they would behave in a hippocampal slice, and the hippocampal slices are behaving almost like in vivo hippocampal cells. So with these cells, this is a less problem. Also, what we can see that we can evoke excitatory synaptic connection or synaptic current, and we can evoke inhibitory. So we have also glutamatergic and GABAergic synapses between the cells. Also, what I will be more interested in, they are interacting with each other. So if one cell is firing action potential, it is inhibiting the firing of the other cell. This is a video how these patterns are forming. This is one cell, this is other cell. They are trying to explore their environment. They are sitting on the hydrophile now. They are sending out processes, and when they are finding some clues, they are exploring the new surface, and when they are finding it appropriate to sit, they are just jumping there, and they are attached to the cell attachment site, and they are sending out the processes on the predetermined pathway. From this video, you can see which is surprising, how cells are exploring their environment and the way how they are moving on the surfaces. We did some experiment. What are those parameters, what we have to create to determine the patterns with the cells? Of course, the surface. We tried out several surfaces, and surface properties definitely influencing cell attachment and cell growth. Also, we tried out different shape, different size for the patterns, and we tried to find the best pattern or pattern geometries to get the best pattern formation. So what are the problems? Okay, we can create cellular patterns. The major problem is from biological part. For these studies, I didn't tell, but we are using serum-free medium. And as I mentioned, serum-free culture is sometimes problematic. In our case, the probability of synapse formation is lower than in a serum-containing culture. So we have a task to optimize the culture conditions to get more physiological behavior of the cells. So there are lots of biological problems. We don't have glial cells in this culture. There are lots of data, the interaction between glial cells and the neurons. We have problems with the longevity of the patterns. Usually, the cells survive very long in vitro conditions. People are talking about 2 years cultures in cortical cultures. The problem is that the surface patterns, the cells tends to overgrow the surface patterns after some time. So the stability of the patterns is definitely not 2 years, maybe a month, but not longer. The other is what is physiological. I was talking about the difference between in vivo, in vitro systems. So are we modeling a real in vivo system with these networks? So in vitro, in vivo, and the other one is biology is redundant. So it's consisting of multiple cells and not single cell networks. The other approach, what we are doing is multiple cell networks. We are modeling, for example, the hippocampus. In the hippocampus, there are well-defined cell layers, and these cell layers are interconnected with monodirectional connections, like the dentate gyrus and the CA3 layer of the hippocampus. What we would like to do, we would like to create cell layers, and we would like to connect these cell layers with monosynaptic one-directional connection. We are trying to design the patterns to create directionality. So exons growing this direction will connect to this layer, but on this layer, the exons will not find the direction back. So hopefully this is one-directional connection. We did some recording, and we showed that the probability of the firing or the firing between these two layers show directionality. So one layer is firing earlier and inducing firing to the other layer according to this pattern. So that is the status of our neuronal engineering. What we are doing now is trying to scale up and trying to do drug screening on synaptic transmission on those cell layers and those cells. The other usage of in vitro systems is toxin detection. Every cell is an ideal biosensor. Question is, what is the information we are able to read out from the cells? Reading out selective information is difficult. Usually what we can read out is whether the cells are alive or not, they are firing action potentials or not. In this case, we are using cardiac cells. It's a cardiac monolayer, cardiac myocytes monolayer on microelectrodes. They are firing regularly, spontaneously. If you are applying drugs, it is changing the firing frequency, and it is changing a bit the size of the extracellular signal. If we would like to step a bit further, we can create patterned cardiac systems. If we are interested how excitation is spread in cardiac monolayers, what is the speed? If we are interested in problems in this excitation spread, we can create a patterned cardiac monolayer line on the electrode, and we can study this phenomenon in a high throughput way. This figure are calcium imaging, so it's not electrophysiology, but optical imaging of calcium waves in cardiac layers in patterns. So we are working on to develop screening systems for cardiac myocytes, which are giving us more information than just firing of cardiac myocytes. We can study recurrent excitation and different physiological properties of cardiac myocytes in these systems. Okay. Another project we are doing, and I'm not talking too much about it, is to recreate the stretch reflex arc. The stretch reflex arc is consisting of a motoneuron, a muscle fiber, and a sensory neuron, and this is a trisynaptic pathway. What we would like to do, take these three cell types and organize them in a working system. I'm not talking too much about it. Dr. Hickman, we'll talk about it later in connection with spinal cord repair. But I would like to say one or two words about system characterization. We're also using mathematical tools to characterize our system, and until we are not able to build them, at least we can simulate them. So we are measuring transfer characteristics of our cells in vitro, and we are simulating the function of the stretch reflex arc if they would be interconnected. So based on real experimental data, we are creating simulations to predict how the system would work. At the same time, we are building the system, and we will compare the results of the simulation and the experiments. For toxin detection, we are also working on the amount of information we are able to gain out of the cells. We are working on optimizing the contact between cells and electrodes, and also trying to do the action potential shape analysis to read out more information about toxin effect. The idea is simple. The drugs are affecting the cells. In many cases, they are affecting ion channels. If they are not affecting ion channels, they may affect second messenger system, which are affecting ion channels. Ion channels are responsible for action potential generation. If I'm affecting ion channels, I can modify the shape of the action potentials. If I have a mathematical model how the action potentials are generated, I may be able to calculate back which ion channel or which system was affected in the cell. I'm doing parameter fitting in a complex model of a cell type. I'm doing recording at the same time. I'm applying a drug, and as the action potential is changing, I'm fitting the model parameters on the new action potential, and I'm trying to draw conclusions which ion channel was affected, basically with drug administration. So, sorry for the confusing. I was talking maybe a bit too much, two different topics and not long enough, or maybe long enough. Basically, the summary is that we are able to build simple in vitro systems. We have some basic tools to do it, but we have lots of other problems, and these problems are originating from biology, that we don't know enough about biology. It is really surprising that how much we know about biological systems and how less it is applicable in our applications. I think every of us is facing this problem, that huge literature and difficult to apply. So we need more experience with our systems, but we have the tools, what we can use for basic research, environmental monitoring, drug screening. I didn't talk too much about in vitro disease models. Dr. Hickman will talk about more in connection with spinal cord repair and different disease models in vitro. I would like to say thank all of my collaborators, Dr. Hickman, our group, and our funding sources, which is mostly NIH and DARPA. Thank you very much.